I am Dr. Aditee Mitra, Research Fellow in the School of Earth and Environmental Sciences in Cardiff University. My research focuses on life in the single largest ecosystem on our planet, the oceans. More specifically, I work on organisms that you may well not even know exists, but they play a critical role in our daily lives. To introduce this subject, I would like to share a video with you. Are you wondering what are Mixoplankton? If they are not plants or animals, what can they be? Just like us, Mixoplankton are eukaryotes. However, they belong to a different kingdom called Protista. And actually, protists comprise by far the majority of all eukaryotes. So they are far more protists than trees and other plants, than mushrooms, than invertebrates including insects, than all animals in the animal kingdom. You may well not have come across the word Mixoplankton in any of your textbooks. You may be wondering why. This is because it is only recently that we have discovered the importance of these organisms in our oceans. You can read more about the history of this exciting story at mixoplankton.org. Let's have a look at Mixoplankton in a bit more detail. First of all, they photosynthesize in a way very similar to land plants using sunlight, carbon dioxide and nutrients, and producing oxygen. 50% of the oxygen that we're breathing in right now comes from marine plankton. However, the way that Mixoplankton eat is certainly not like any land animals. So let's go and have a look at how they eat. In this video, we see three smaller Mixoplankton of the same species ganging up on one larger, different Mixoplankton species. They're eating by engulfing their prey. This is a time-lapse video. The feeding event would take a good few minutes in reality. Obviously, there are some part of their prey the Mixoplankton do not like, as at the end they leave a bit behind. So what does Mixoplankton research entail? Having lots and lots of fun. We go out to sea to look for plankton. We use nets of different sizes to collect different types of plankton. Here you can see a big net being deployed from the side of a big ship. And here you can see a much smaller net that we deployed from the side of a small boat on a day sampling trip. Once we have our species, then we look under the microscope. We try to identify as many plankton species as we can. From these surveys, we hope to have an idea of what species are present in different parts of the oceans, at what time of the year, and how many. The next step is to try and build a conceptual food web model. We do this to understand how it all plugs together, how the system works. This is a bit like the blue whale food chain that you saw earlier. Following this, we carry out various experiments in the laboratory. We measure things like how fast they grow, how fast they move, what do they eat, what do they not eat, what nutrients do they use, and who eats them. We use data from these experiments to explain the different biological functions. Specifically, we use these to make mathematical models. So why make mathematical models? Mathematical models are very important for everyone, not just scientists. For example, the weather forecast is dependent on the operations of several models used by the Met Office. Climate change projections also make extensive use of models. All these models use descriptions of plankton. So getting the plankton models right is very, very important. And this is one of the things that we do in Cardiff University. Mathematical models are very much like computer games. So in my job, I get to design computer games and play with them as well. I develop plankton models of different complexities. I plug them together to build food webs. I then run simulation experiments. I investigate what makes marine life thrive or not.
So now we're going to build a simple food web model. For this, we're going to use the plankton food web that we saw at the base of the blue whale food chain. So let's go! We use a software called PowerSim Studio to build our models. The very first thing which one has to decide when making a model is what currency to use. In this instance, we're going to be using a nitrogen-based model. So here we go. We're starting with a new sheet of paper, new sheet on the screen. We're going to first introduce our players. So we have got the nutrients here. And we're going to, it's a question mark, so we're going to define how much nutrients are being used. And let's put a nice picture to show us what it is. Next, we're going to define our mixoplankton. So here we go. So we've got mixoplankton here. Let's call it mixoplankton. Again, we go in, we define how much. So these rectangles are called levels, and they represent the biomass of the players in a system. So now we have got the mixoplankton biomass in nitrogen units and the nutrients. So they're going to take up, so we're going to use these things called flows. So nutrients are going to be taken up by a mixoplankton. Here we go. And this is where the in this equation goes. So let's first call it, it's taking up nutrients by mixo up. Okay, we need some constants. So it's going to take mixoplankton, it's going to take up nutrients at a certain rate. So let's call it mixo. Okay, so let's call it mixo up. So that's mixo uptake rate. And then this one, we'll call it, to define an equation, we need something called a half saturation constant. So let's call this okay. These constant values are the ones which come from different lab or field experiments. So we're going to put in 7, Okay, and we're going to put in a value. That's why there's a question mark. Let's put this in. Now we're going to link all this up. So the Mixoplankton are taking up the nutrients. So we will need the Mixoplankton, the nutrients, the Mixoplankton, and these two constants. And now the time to put the equations. So we've got nutrients. So it's going to be, let's start with the Mixoplankton times the uptake rate, which is this, times, this is where we use which is called a rectangular hyperbola, but this equation can be of different forms, and what form it is depends on what we understand about how these organisms work. So this takes us back to those physiological experiments we talked about earlier. So here we go. So we've got it. So we've got nutrients being taken out of Mixoplankton. Now we want to check if it's working. So let's make a graph. Now we put the nutrients in here. Here we go. And we put Mixoplankton and let's run. There we go. So what we can see is the nutrients run out and the Mixoplankton grow. So we are going to next add a krill. Let's put a krill in. There we go. So we put krill and we put in a number. Let's also find a nice picture so we know it's there. I think that's it. Here we go. Okay, and it's a krill biomass. We're going to link it up. We need, so the Mixoplankton, it's going to be graced Mixoplankton, so there's a K, aim, predation. Here we go. So again, we will need two constants. So let's have now K, M, and so let's call this Km grace constant, and we call this KK grace constant. Go, let's put some numbers in again. Let's put same. Move this across. We need to link them all up again. 1, Mixoplankton 2, grazing function, half saturation constant, and then we need to go and write in our equation. So let's say krill time grazing times Mixoplankton divided by. So you can see why we would like to use short names or even symbols, because otherwise the equations, as you keep making more and more complex models, get more and more complex. Here we go. And let's see, let's add this on. Let's go back and let's run. And there we go. So we now have got, what we can see here is that the krills are taking over. So this, then we go on to build, add more and more components. So I would love to show you the whole thing, but I can't because of time constraints. But here is what we would do next. So from the krill, we would have an output, the excretion. This is associated with respiration and also digestion. So they would go back into the nutrient pool. We will also have the krill, just like any animal does poos as well. So that will go and join the fecal material box. And then we have the bacteria, which are going to be using the fecal material, taking it up, and then they produce nitrogen as their products of digestion, end products of digestion, and they go and join the DIN pool. And there's, of course, the bacterial grazing by the Mixoplankton. So this has got a very simple food web of the Mixoplankton krill at the base of the blue whale food chain. So we're going to run this model now. So the other thing which I've got here is a switch, which switches between either the Mixoplankton just doing photosynthesis or the Mixoplankton doing photosynthesis and eating. So we're going to start off with it just doing photosynthesis on 0. So let's run it. Here we go. So we see one set of dynamics. Now we're going to switch to when the Mixoplankton is photosynthesizing and eating. And let's run again. And what you can see here is a change in the dynamics, and that is what we look at with models, is how just changing something as simple as this or something more complex changes the entire dynamics of the food web. So from this we will go on to add other impacts like the movement, quality of food, and other different components, players in the food web, to find out how changes in the environment, whether it's temperature, salinity, etc., impacts our marine food web, the marine ocean life.
And that brings us to the end of this presentation. I hope you enjoyed learning about Mixoplankton. How marine life, including the largest animal on our planet, depend on these amazing microscopic organisms. And how we integrate mathematics with biology and ecology to study life in our oceans. If you wish to find out more, please visit our webpage, where we have lots of information, including blogs of students working on Mixoplankton. Thank you for your attention.